Hey guys, it's Ron of the Rotifera here, and I will be filming today a Draw My Life video. A little disclaimer, this video is sponsored by Loyola Marymount University Biology Lab 111, Section 15. I was born on December 5th, 2016, making me 3 days old and fully grown when you are watching this video. I don't know how much I weighed because I am microscopic, and I do not know exactly who my parents are, but I do know that I was either born through sexual transmission with a mother and father, or I was born through parthenogenesis, with only a mother asexually. During the first 2 days of my life into adulthood, I grew to have a corona, which helps with my motility and feeding, a mouth with a jaw behind it, a brain, stomach, intestine, anus, toe, and a foot. I am also known for being protostome, meaning that my mouth was formed before my anus. Other quick fun facts about me is that I am bilaterally symmetric. I have two tissues. My body cavity is pseudocoelomate. I am externally segmented. I have an external layer of chitin called lorica. I do not have a circulatory system, nor do I have a respiratory system. I live in a freshwater environment. I'm known for eating bacteria, protista, and I tend to be parasitic. I am also deathly afraid of shrimp and crab. The cool thing about being me is that I have thousands of brothers and sisters, that we have 3 different classes to be identified with, which are Monongananta, Podolidia, and Sasanidia. I generally spend my day swimming or crawling around with other rotifera like me, just helping to stabilize organic wastes, stimulate microfloral activity and decomposition, enhance oxygen penetration, and also help the recycling mineral nutrients. When analyzing my 16S rRNA phylogenetic tree, I found myself to be closely related to the mollusca phylum, my sister taxa. We share a common ancestor. I always knew I had a lot in common with Sally the squid. The mollusca and I are most closely related to the arthropods like the crayfish. The scientists in biology labs studied the morphological characteristics of the same 9 organisms to create a data matrix based on morphology, and I am in the sister taxa with the echinoderm phylum, in particular sea cucumbers. This means that the echinoderms and I share a common ancestor. The ancestor that we share is the phylum Neotriploblastica. The sea cucumber and I have very different morphological traits. Although there are many differences about us, the traits that we do have in common are ones that we don't share with anybody else in the tree. When comparing the 16S rRNA and the data matrix, there are some distinct differences. The 16S rRNA tree was created based on genetic similarities between the phylums, whereas the DNA matrix is focused solely on morphological characteristics of the different phylums. This one was found in the journal Molecular Evidence for Acanthocephala as a subtaxon of Porifera, written by James Gray and Thomas Neer. This tree looked at more than 9 phyla with a total of 16 individual phyla. This, similar to the 2 trees we've already looked at, the Cnidarians and the Porifera are close to the root. The Porifera is the outgroup once again. In this tree, I am in a sister group with Acanthocephala, which is a parasitic worm similar to me. This phylum was not taken into consideration with the 2 former trees, where I was in sister taxa with Echinodermata and Mollusca. As you can see in this tree, I am still very close to the Mollusca phylum, which is very similar to the first tree, which was constructed by studying DNA code. The only part of me that can possibly fossilize is my jaw. My jaw is so small that detection is nearly impossible. The oldest discovered fossils of Rotifera were found in Dominican amber and are found to date back to the Eocene period, 55.8 million to 33.9 million years ago. We can hypothesize that the rotifers originated as late as 55.8 million years ago. The study by Gray and Neer included a molecular analysis of rotifers and Acanthocephala. In this topology, they found that the molecular analyses included only 28 substitutions. The Acanthocephala and I share the common ancestor of Syndermata. The one trait that set me apart from all the phylum that originated from Syndermata is my corona, or wheel organ. My corona is located in the cephalic region, which is used for locomotion and food gathering. The difference between the journal's phylogenetic tree and the biolab group's phylogenetic tree is that the journal used molecular differences and the biology lab group used morphological differences. If the molecular differences are used in the phylogenetic tree, the tree will be more accurate.